Dennis Desjardin, who's a good friend of this club, is here tonight to share 40 years of discovery, new species, new insights, and new directions. Dr. Brian Perry, a former student who may know him best, is also here with us tonight, who's also a very good friend of this club, to introduce Dennis. Brian.
Hey, everyone. Hello, everyone in Zoom land. Yeah, as Mike said, it's an absolute pleasure to get to introduce your mentor when you're giving a talk. And I had to, in Dennis's case, I had to actually take some notes. I had to write down all of Dennis's accomplishments and things like that. So just to give you a little bit of a backstory with us, Dennis is really the reason that I'm standing in front of you here tonight. So when I was an undergraduate at San Francisco State University, taking my second semester general biology class, he came in and gave a guest lecture on fungal biology and blew my mind. Completely blew me away. I could not wait until I had taken all the prereqs and I got to enroll in his upper division of mycology courses. When I did that, by the end of the first month, I knew this is what I wanted to do. So we were just talking about it a few minutes ago, just through a series of like random chances, I ended up becoming a mycologist, going on, getting a PhD, and now I do similar work to him over at Cal State East Bay instead of San Francisco State. And so someone once said to me, like, what if you had skipped class that day? You'd never heard Dennis lecture. Who knows? I'll probably have a lot of money or do something else. Standing my professor studying mushrooms. What are you going to do? All right. But, Dennis has been an incredible mentor. He really, he gave me an incredible number of opportunities and continually fostered my interest and my passion in the subject. And it's something that I've really tried to emulate with my own students at my own university. So it's been a fun road, and I hope I can, sort of live up to the sort of mentor that he was for me. So to tell you a little bit about Dennis, he's had a very interesting background and interesting path as well. So Dennis went to San Jose State from 1968 to 71, year I was born, by the way. He was a math major, and he also had a minor in Asian philosophy and a minor in music, but he didn't finish. He left to pursue music. And so for about 10 years, you played in various bands around the Bay Area and had a very prolific music career during that period. And then in about 1981, he decided it was time to go back to school. He had collected mushrooms with his parents and grandparents growing up, and he decided that he wanted to study mycology. And it just so happened that there was a mycologist by the name of Dr. Harry Thiers at San Francisco State University. So Dennis completed his undergraduate degree. He got a BS in botany, correct? And then he completed a master's degree in 1985 in ecology and systematic biology, working with Dr. Harry Thiers. Right after that, he went straight into a PhD program at the University of Tennessee to work with Ron Peterson, where he continued his work on the genus Marasmius, if you know that. He is the world's authority on that group of mushrooms, and did some incredible work there for his PhD on that group. And then after that, he did a one-year stint at Oberlin College in Ohio. That was his first faculty position. And then in 1990, he came back to SF State and replaced his mentor as he stepped down and retired and became emeritus faculty himself. And so Dennis was at San Francisco State from 1990 up until 2021, right? So for 31 years, he was there, retired in 21. And now he is emeritus faculty, more busy than ever, I think, doing many, many different things, including research. And so during that period, this is what I had to take notes on. Dennis mentored 24 master's degree students, a number of which are in the audience tonight, and 5 PhD students, okay? During his career, he has he has published over 160 peer-reviewed scientific papers and co-authored 2 field guides to mushrooms, one of Hawaii and one of California. Dennis he's one of the few mycologists in the world that has won 3 of the most prestigious awards from the Mycological Society of America. He's the winner of the Alexopoulos Prize and the Weston Teaching Award. He got both of those in the same year. And then just 2 years ago, he was awarded the Distinguished Mycologist Award. And so a highly decorated mycologist in the Mycological Society of America, well-respected and cherished by his students and coworkers. Let's see what else. Finally, the important stuff. He has described 302 species that are new to science, described 7 new genera, and is responsible for countless combinations, like changing the names of things that make all of you very angry. He's done lots of that as well. He has done field work and mycological studies, obviously in the United States and the continental USA, but also in the Hawaiian Islands, Indonesia, Micronesia, Thailand, Malaysia, West Africa, and most recently, Brazil. Okay, so he's done a lot. He's got an amazing background, and he's going to tell us all about that tonight. So please help me welcome Dr. Desjardin. All right. Recording back to you. All right, very good. Back to screen share. Hold on. Slideshow. All right.
Technical stuff here. Well, thank you, Brian, for that interesting introduction. Thank you all for having me here. The Mycological Society of San Francisco has a big place in my heart. I actually joined the society in 1980. So I've been with you guys for 44 years now. Actually, I was the first librarian of the MSSF as well, and it was my job to organize those books and keep a list of it. And this was before computers as well, so a long time ago. Harry Thiers was the scientific advisor at that time. And then when I came back to San Francisco State, I became the scientific advisor as well. So I've been your scientific advisor for 30 some odd years as well. So it's been a lot of fun. We've had a lot of good times together, and I've had a lot of chance to actually talk to you a little bit. So I'm going to do something a little different today. I figured that I've been doing this long enough that I can talk a little bit about what I've been doing for the last 40 years. So really what I want to do tonight is give you a little bit, and Brian already covered my first two slides, so I can probably just zip right through those. What I want to do is tell you a little bit about some really interesting species that we found, species that kind of changed a little bit of our concepts of both morphology and evolution. And then I want to talk a little bit about some of the various phenomena that were very interesting. I want to talk very briefly about bioluminescent fungi. I don't want to go too much into that, although probably more than you want to hear, because Brian gave you a wonderful talk about bioluminescent fungi this last season. And then I have a reputation for trying to put fun in fungi. So I want to talk to you a little bit about some of the fun we've had with fungi. And then I'm going to finish it up with one slide, talking about the new directions where I've headed. I retired in 2021 and lasted about 3-4 months. And then I got headhunted and moving into a different direction. So I'm doing a bunch of other stuff now. So with that in mind, it all began here. Anyone know where this is? Northern California.
Yeah, it's Del Norte County.
Del Norte County, that's Crescent City. So it all began there. Get this started here. And it began with this beautiful lady over here. This is my mother. And that's what I looked like when I was playing music back in the 70s. Definitely a hippie back then. This is my grandparents, my mom's parents, Louis and Maria Tozio. They immigrated from Switzerland in 1920 and started collecting porcini, meadow mushrooms, and chanterelles in Del Norte County under the spruce trees. So when my mom was very, very young, she went out in the woods to collect those 3 edibles. And of course, everything else was deadly poisonous and you're not supposed to touch it. Right? And then when I was 3, Mom and Grandpa took me out to start collecting mushrooms in Northern California and got me interested in it. So I've been collecting for well over 70 years of the mushrooms for the table. As an inquisitive young person, I realized, how can these 3 be edible, but these other hundreds that we're seeing around here not be edible? So I literally started studying mushrooms when I was very young. And this was back in the late 50s and 60s when there weren't any North American field guides, other than one that was written in 1900 by Atkinson that nobody had, until Orson Miller came around in 1976 and wrote Mushrooms in North America. And it's a big doorstop book, much like my California mushrooms book, weighed about 5 pounds. That came out in 1976, and that was a book everybody used. And most of the mushrooms in it were Eastern North America, but we used them to identify mushrooms in California, which we know doesn't work very well now. But anyway, so I got into it there and decided after trying to play music, actually playing music professionally for a long time in the Bay Area, that I wasn't going to make enough money to have a good life. Decided, as Brian said, to go back to school. So I went to San Francisco State to study with this gentleman here, Dr. Harry Thiers. And then I went to Knoxville to study with Ron Peterson here. I always believed in the guru-disciple approach to learning anything. You find the best people, and then you sit at their elbows and learn what you can from them. Doesn't matter what the university is. If the best person is there, you go there and you study from them, and then the world opens up for you. And then I came back to San Francisco State and took over for Harry Thiers when he retired. I just put this up. Brian already mentioned a little bit, but here's a map of the world. The circled areas are areas where I have been able to either spend a lot of time in the field doing field research, or I have had a lot of time to study the fungi of that area, borrowing and studying the type specimens to get an idea for what the diversity was there. And up at the top are some of the major projects that we've had in my lab around the world to try and document the diversity of mushrooms, and particularly important because we're losing so many habitats around the world that if we don't document what's there now, we will never know what was there. So I was lucky enough to receive funding mainly from the National Science Foundation, but from all these other funding agencies around here to allow me to literally just walk in the woods and pick mushrooms. Of course, you had to do something with them after you picked them, but basically go around these wonderful and mostly in underexplored Asian and African and Brazilian tropical forests. So that allowed me to spend a lot of time to actually find these mushrooms, study them, later on in my career, sequence the DNA, run phylogenetic studies, and then place all of this understanding of diversity into an evolutionary context. So as I said, what I want to do is just kind of take you through a few interesting ones to show you some bizarre fungi that you've probably never seen before and talk to you a little bit about why they're interesting. So we'll start off with something like this. So this little guy here on the left, this is Gliocephala aquatica. This was really the first mushroom-forming basidiomycete that was described that is aquatic, growing in fresh water, but not just aquatic. It grows under ice in the winter in fruits on sedge. And here's where we collected this. Was in Argentina at this lake here, Lake Zeta. And you can see the ice on it in the winter. Around the margin here is where the sedges are. And this little guy right here, fruits on those sedges. This is the underside of the mushroom. The stem is on the side. And here it doesn't have any gills. It's a smooth undersurface. The spores are produced right there. The spores are very, very, very long and skinny, almost like a hypha itself. And it forcibly discharges the spores into the water, and then they float around and find other sedges. So it was, at this time, nobody really thought that mushrooms could actually form and grow underwater. It was about 15 years later that the friends in Oregon discovered a Satharella species. They described as Satharella aquatica, which is a little gilled mushroom that grows in the Rogue River on sticks underwater, fruits underwater. So this was really interesting for us to actually find something that is psychrophilic, which means cold-loving, and also aquatic. And then a few years later, we found this one, and this is a Physolacria mapoensis. This mushroom has a central stem, it has a round head, and the spores are produced all over the surface of that round head. Now, I'm going to go back just for a second so you can see the scale bar. That's 2 millimeters. So these are small mushrooms. So I'll show you the scale bar on this. That's a 0.5 millimeter. So this whole thing is only about 2 millimeters, maybe 2.5 millimeters tall. Very, very tiny. This grows in salt water. It grows on the base of mangrove trees that inundate daily, covers them daily. And we were able to grow this in petri dishes with a 33% salinity, which is far more salty than the water that it grows in. Very, very few mushrooms, if any, are capable of growing in salt water or in any saline soil even. But this really interesting little species, we discovered in Hong Kong. And here's a real bizarre one. This is an inside-out mushroom. Again, a scale bar, 2 millimeters. That's just very tiny little mushrooms. What happens with this one is it looks like when it's young. I don't have the youngest stages of it showing here. When it's young, it looks like a martini glass. So you've probably seen some ascomyces, little cup fungi that have a stem that look like a martini glass. And it's inside the cup that the spores are formed. And in ascomyces, they forcibly discharge their spores into the air quite a ways away, and then the wind picks up the spores and carries them away. And so that works well to have your spore-producing structures facing up if you're an ascomycete. It doesn't do well if you're a basidiomycete to produce your basidia facing upward, because when they discharge their spores, they discharge them about 20 to 30 microns. Very, very short distance. And typically basidiomycete spores, like all the mushrooms that you're collecting, they are produced on the underside of some sort of a cap. So the spore and the basidia that produce the spores are parallel to the ground. So when it shoots off the spores just a few microns, they drop down by gravity, and then the wind picks them up and blows them away. So if you're going to be shaped like a martini glass when you're young, how are you going to get the spores out of that cup if they're forcibly discharged? Well, what this fungus does is it grows up, and the stem is in the middle of the cup, and then the cap turns upside down, and now the spores are inside the cup, and the cup's upside down. So this is a mushroom that has a stem on top of its head, and it has to curl over in order to discharge its spores. We first found this one in Australia, and then we found it more recently in both Thailand and Malaysia. This was a new genus when we found it. And we've got a lot of molecular data on it now. Here's another really interesting one. And this is one that we first found in Thailand. Now, if you look at the mushroom here on the left, when I first saw this, I thought it was a loofah sponge. If you guys know what a loofah sponge is. It looked just like a sponge. It's about the size of an orange. I picked it up, and it was soft and rubbery. I squeezed it, and the water rang out of it. And then I let go, and it puffed back up to its natural size. You don't do that with mushrooms. You squeeze them and they break all up. Well, this guy was just like a sponge. And when I studied it, I realized it as well represented a new genus. So since it's like a sponge, I call the genus Spongiforma. We first found it in Thailand. It has no cap, it has no stem. It's just this big, gnarly sponge-like mass. It grows above the ground and is mycorrhizal with dipterocarps, which is a very important mycorrhizal group of trees in Asia. We did the molecular biology on this, and we realized that it was closely related to a bolete. So this is basically like the sponge of a bolete without the cap and the stem or anything else. And it just grows above the ground, just in this sponge form. 3 years after we described Spongiforma thailandica, this genus here, Borophytus, was described from Bangladesh. And as it turns out, the closest relative of our Spongiforma from Thailand is this bolete. Now, it's a good bolete from Bangladesh. Interestingly enough, just a couple years ago, Borophytus was reported from Thailand. From northern Thailand, not too far away from where we actually found Spongiforma. So probably this thing evolved in this area of Southeast Asia. And when you look at it, this sponge is more closely related to this little bolete than that bolete is to any other boletes. So those sorts of things are just kind of weird. And then we found another one. Actually, Tom Bruns and Kabir Pei. Tom was at Berkeley, Kabir Pei is at Stanford. They were doing research in Borneo, and they were studying mycorrhizal fungi in Borneo, and they came across this little tiny sponge that's bright orange, and they sent it to me and said, we think this is in your same genus, that Spongiforma that you found in Thailand. And sure enough, we did a bunch of work on it. We realized that it really was. And the thing is that it's the one we found from Thailand smelled like coal tar, kind of like asphalt, really awful smell. This one is very fruity, kind of smells like a pineapple. And here's some SEMs of the spores. The spores look like a pineapple. Well, we figured SpongeBob lives in a pineapple under the sea, and he's a sponge. So we named this after SpongeBob SquarePants. So this fungus is Spongiforma squarepantsii. What can I say? Well, it was so important that actually there is a group of scientists in the United States, part of the International Institute for Species Exploration. And every year they look at all new species across all life forms. And there are about between 3,000 and 3,500 new species of life, from bacteria to plants to all animals and fungi, that are described every year. They look at all those species in a given year, and then they choose the top 10 that they think are the coolest species found that year. Well, in 2012, Spongiformis squarepansii was one of the top 10 species, and we got the top 10 award. So that was pretty cool. And then back here in the West Coast, here is a truffle-like fungus. Okay, this is an underground fungus. Often you guys are all calling them truffles. True truffles are ascomycetes. This is a basidiomycete truffle. It grows under pine trees in the Sierra Nevada. Now, if I was in my right mind back in 2003 and what everybody was telling me, if you look at this thing, it actually has all the characteristics of a genus that's called Zelleromyces. And Zelleromyces is basically an underground Lactarius. This one has red latex. If you know Lactarius deliciosus or Lactarius rubrilacteus, deliciosus has an orange latex. Rubrilacteus here in California has a red latex. This one exudes a red latex, and it stains green, just like Deliciosa group stains green as well. And it actually forcibly discharges its spores, even though it grows underground. And most of those don't do that. We found that by cutting it, putting it on a piece of paper, and it dropped a beautiful spore print, interestingly enough. So basically, this thing is a Lactarius, but it lost the stem. There's the stem right there. It's this little bit of a columella in the middle. It lost the stem, it lost the gills, it lost the cap, it lost the peridium, everything else, and it just crammed itself together like a bunch of mixed up gills, shoved itself underground, is living happily in the Sierra Nevada underground. What was interesting about this one was I knew that it was a Lactarius, an Hypogeus lactarius, but at the time when we described this in 2003, any of these underground ones were always described in the genus of Hypogeus fungi that everybody thought they belonged to. So everybody told me, my colleagues, particularly the people that study these, like Jim Trappy up at Oregon State, you got to call that thing a Zelleromyces. And I said, no, it's an underground Lactarius. So this was one of the first, this is actually the first Lactarius that was described in the guild genus, but it doesn't have gills and it's underground. So this kind of started a little bit of a trend. And as it turns out, we didn't know it at the time, but there is a Lactarius in the Rocky Mountains. This is called Lactarius barosii with green stains at maturity and red latex. That is the sister species to this. So this underground Lactarius is more closely related to this above ground Lactarius than this red latex Lactarius barosii is related to Lactarius rubrilacteus. So again, interesting. You can change your morphology with probably just a few gene changes. And the adaptation is to move out of this dry, colder environment and go underground where it can retain water more and avoid some of the cold and freezing temperatures we get in the mountains during the winter and survive happily underground. So those were some ways that fungi have invented in being different morphologies for being a different mushroom. What I want to show you here are some interesting ways that some common genera, or at least this one genus, Mycena, which Brian knows pretty well. Brian and I have been working on Mycena for many years now. Different ways that they've invented to be productive. Okay, so as you all know, these little mycenas, they produce gills underneath the cap here. They produce sexual spores underneath those gills. And just like any good mushroom, they discharge those spores into the air. And that's good reproduction, a lot of genetic variability because of the sexual meiosis and all of that in sexual reproduction. This group of little mycenas you can see here, here's a common one here in California. Both of these are common in California. If you look at the cap here, all those little granules, they look like sugar granules, which is why they were placed and given this name in section saccharifery, like saccharin, like sugar. Okay, if you look at those little granules there, and now look at these over here, you take those granules and you pile them all up and make a little spine, and you have this beautiful little top notch here. And same way on this one here. Well, as it turns out, all of the cells that form these granules and these little spines on top are actually a universal veil. So this is the first time that anyone had really described a universal veil being present in a mycena. And most of you probably know mycenas. Well, all of them in this group here produce this interesting universal veil. This interesting universal veil is formed from special cells called acanthocysts and kerocytes. So just to show you kind of what they are, here's a couple drawings of them. And here's one of those spines. It's formed of chains, as you see I've drawn here, chains of these cells. Each of those cells has little tiny spines and big spines on top of them. And over here you see them as well. So these thick-walled ones are called the kerocytes, and the little ones here have those little tiny spines all over them are called acanthocysts. As it turns out, this universe, and here they are under the microscope. So the pileus surface is this layer right in there. Sorry, on Zoom you can't see that probably. And up at the top there, this is a universal veil. What I did was each of these cells will disarticulate. They will break apart from each other. When you plate them out on some good agar, each of those cells will germinate and form hyphae. So these are asexual spores. They're asexual reproductive structures. And so you have this universal veil that when it rains or when insects get these little spines caught on them, they can carry these cells around and they can clone the species with this asexual reproduction. So this is, again, kind of the first time you've ever seen a universal veil acting as an asexual propagule and doing so on these beautiful little mycenas. Really, really fantastic. It's a great way. So these are smart little guys. They reproduce sexually with all the genetic variability, and they reproduce asexually so they can spread themselves and clone themselves. Incredible. As I said, I wanted to touch a little bit on this because this is what we've been focusing, Brian and I and our colleague from Brazil, Cassius Devani, have been working on for 15 years now, I guess. For me, it all started with this guy over here on the left. This is Geranium viridescens. My friend Cassius was doing some research. He's a chemist, physical chemist, and he did his PhD on the chemistry of light sticks. And he did his PhD on the chemistry of fireflies as well. So he was out in the woods in Brazil collecting, and he's camping out overnight, and they found through their hikes this beautiful little mushroom here, Geranium viridescens. And he looked online to find out who might know something about the mushrooms of Brazil, found my name, sent me an email saying, hey, we've got this really cool bioluminescent mushroom down here. Do you know what it is? I said, send it to me. So we worked it up, and as it turned out to be a new species, and not only a new species, but a new lineage of bioluminescent fungi. We knew that the armillarias were luminescent, their mycelium is. We knew jack-o'-lantern mushrooms, Omphalodus, was luminescent. We knew Panellus stypticus was luminescent. So we've known for a long time that there were three lineages. Well, then here comes along this guy. It's like, wow, that's a whole new lineage. And nobody knew that anything related to this actually was luminescent at all. So we published this. Cassius liked it because I wrote the paper and he was a co-author on it. All I had to do was mail me the mushroom. So that worked out very well for us. And he said, hey, we've got at least 8 more down here. Are you interested? I said, send me a flight ticket. So he did, and that started our collaboration over the last 15 years. Over that period of time, we discovered 20 more bioluminescent species in Brazil alone. And subsequent to that, dozens more all over the world. And these are just a couple. We made the cover of Mycology in a couple different times. Here's what we know about them now. There are 132 species now. Back in 2008, I wrote a paper, kind of a review of what was known at that time on the bioluminescent mushrooms of the world. And there were only 64 known in 2008. So 16 years later, we've more than doubled that now, mostly with our work and some other people's work around the world, to 132 species. And this is just kind of showing you where they are. You'll notice over here in this little bar graph, we now recognize 5 lineages. I'll say more about that in a minute. But you see on the right here, that tall with 96 species, that's the genus Mycena. Most of them are in Mycena, and it just so happens that we were kind of experts on Mycena. So that worked out well for us. And here are just a few. 73% of all the known bioluminescent mushrooms belong in the genus Mycena or related to Mycena in the Mycenaceae. Here are some of the new ones that we described from Malaysia. This one right here is Mycena lucentipes. That was a new species we described from Brazil. That's Taylor Lockwood's wonderful photograph there. So you take a picture in the dark and then you flash it at the very end. You can get both the light coming off the stems and you can also see the color of the cap. So really beautiful photograph. What Taylor did was he heard that the US Postal Service was going to have a series of stamps called bioluminescent life. So he sent his photograph off and convinced the US Postal Service that they should include, hell, you got fireflies for terrestrial. Why don't you have a bioluminescent mushroom? So as it turns out, Taylor's photograph, a different one than this, but of our species, Mycena lucentipes, is the first mushroom ever that is the focus of the US postage stamp. And there it is. You can buy those. They're still available. They were published in 2018. And all of the rest are aquatic except for the firefly down here, because as you know, about 80% of everything below a couple miles in the ocean is bioluminescent. So there's lots and lots of aquatic bioluminescence, but very few terrestrial. So that was fun. And of course, we have to go back here. One of our species, Mycena luxiterna, also was chosen as one of the top 10 new species. And this was in 2011. And the name luxiterna, I came up with that because one Christmas, a friend of mine was singing part of Mozart's Requiem at a church in San Francisco. So I went and we're singing, and you have the lyrics, and we're singing along to Mozart's Requiem. And we get to the part where the lyrics in Communal are Lux Aeterna Lucit Eis Domini. And I'm going, oh, the eternal light of God. That'd make a great name for a bioluminescent mushroom. So Lux Aeterna means eternal light. The second line from that is Lux Perpetua, da-da-da-da-da. So we described a second species, Lux Perpetua, the perpetual light. So we got two mushrooms now, thanks to Mozart. You got to get your inspiration wherever you can. And Brian mentioned this in his talk as well. We had a new discovery just last year, and that was this a fifth lineage, and that's the Eocyphella lineage. This is a new genus that we described. I want you to see the size of this one as well. It's kind of hard to see, but here is, these are millimeters, and it's less than, the entire fruiting body is less than 1 millimeter in diameter. And you can see that they're 500 microns. These are little upside down urns. The spores are produced in the inside, they hang down, and they glow. The light is given off around the margin, the opening of the urn. And they have little water droplets that accumulate on there, which magnify the light. And so they act like little lenses. Really cool. So what we tried to do was, we knew that there were these 4 lineages when we first started doing this work. And the question automatically comes, well, did this luminescence in the mushrooms evolve more than once? There are 4 distantly related groups of mushrooms that emit light. Was this one early evolution of luminescence and then lost through almost all of the lineages of mushrooms, but retained in a few of them? Or did this evolve independently at least 4 different times, now 5 different times? So to address that, what we did was we extracted with a hot tinctures the substrate. It's called a luciferin. And with cold solution, we extracted a soup of enzymes, because enzymes are heat intolerant. And then we did that from Armillaria lineage, the Omphalodus lineage, the Mycena, the lucentipes lineage, and the Mycena lineage. We extracted the substrate, and we extracted the enzymes, and then we cross-reacted them in all possible combinations. And we found out that whenever we reacted the enzyme with the substrate from any of the lineages, that cross-reacted and gave off light. And when we reacted any of those with a control that did not produce light, they gave off no light. So it suggests that each of those lineages produced the same substrate and the same enzymes to give off light, suggesting that there was a single ancient evolution of bioluminescence in the fungi. At the same time, Cassius was working really hard to try and isolate the substrate and to isolate the enzymes. And we got very, very close over about 10 years of work in isolating the substrate, published several papers on that, but didn't quite know exactly what that substrate was. And then the Russians came along and read our papers and used our techniques, but came at it from the back door. We were busting it, with a hammer. They came with a scalpel in the back door and figured out what the substrate was. And then we figured we would collude with the Russians. So we started colluding with the Russians and then figured out what the enzyme was in conjunction with the Russians as well. And this was then published in 2018. And there's the complete cycle that I won't go into for. It's now very well known and one of the most well-known biochemical pathways for luminescence in any of the organisms. So that was fun. And then we've been working on the phylogenies, particularly with Brian. These are some of Brian's trees here to show you that now there's good solid evidence that bioluminescence in the fungi evolved only once. There are 5 separate lineages, and it evolved way back early in the origin of the agaricales, the group of mushroom-forming fungi, and then was subsequently lost or inhibited in most of the lineages of yield mushrooms, but is retained in these 5 select lineages. And there's just a tree showing you where those lineages are. And this is going to be published this soon. It's been in review for a while now, so hopefully that will come out as well. And then I want to mention as well about the function of bioluminescence, because that's always the question. Why the hell do these things glow? Okay, they're glowing 24 hours a day. So they're emitting light 24 hours a day. That has to be energy consumptive, right? So is there a reason for why they're doing that? So all of the luminescent fungi emit light in this wavelength. It's this yellowish green light. As I said, they emit it 24 hours a day, as long as they're actively growing. If they dry out, they quit emitting light. They do so in a circadian rhythm. Now, unfortunately, when the popular media picked up on this in the paper that was published on this, they said, oh, the mushrooms only glow in the dark. No, they don't only glow in the dark. They glow all the time. You can just see them in the dark because the rest of your light waves obfuscate most of the light that you can see. But they form a circadian rhythm. They emit more light at nighttime and less light during the day in a daily diurnal cycle. It's really amazing that they do this. They require water and oxygen in order to do it. If you reduce either of those two, it quenches the light. There are different phases that emit the light. Some species of mushrooms, only the mycelium glows. Other species, the mushrooms glow. And others, both the mycelium and the mushrooms emit the light. And then for those that have mushrooms that emit light, there are different parts of the mushroom that glow. Maybe just the stem, maybe just the gills, maybe the pileus, some combination of all of those. So then we have to ask the question, why do they emit light in the first place? Well, there are 3 hypotheses that we have addressed. And we have actually data on all 3 of these. I want to attack the first one first. The first hypothesis is that these mushrooms, they're emitting more light at nighttime. So maybe they're attracting nocturnal animals of some sort, insects or other small animals that see them, that come to them to feed or to lay their eggs on them so their offspring have something. And that's just a good food source or a good food source for their offspring. And then in doing so, the mushroom also sporulates more at nighttime than it does during the day. It's also often more humid at nighttime. So as they're there coming to the mushroom, the spores land on the insect's body and then aids in spore dispersal. Well, I'll tell you is where we find the most bioluminescent mushrooms are in closed canopy, tropical forests where very little wind penetrates. So in an area where you don't get a lot of wind for wind dispersal, maybe insects can actually disperse your spores better. There's maybe some selective advantage to that. Okay, so that's one hypothesis. The other hypothesis is that they attract the predators of the prey that feed on the mushrooms. So you can use the number 1 hypothesis well if it's the fruiting bodies that are glowing. If it's the mycelium that's glowing, the mycelium is not producing any spores. Why would you glow and attract some insect to come to you and eat you? Because that's all they're going to do when they find your mycelium, because you're standing out there glowing, they're going to eat you. Well, maybe the fungus is glowing to attract the predators of the insects that eat them. Okay, we actually, I'm not going to talk about that tonight or show you, but we actually have some videos of spiders that come to mushrooms that glow at night and the mycelium that glows at night and hang around and wait for little insects to come, and then they grab them and they eat them. We got some beautiful little videos of that. It's pretty cool. And then the last one, which is not as attractive, but probably more elegant, is that the fact that they're emitting light really has almost no selective advantage of, ecologically or for animals or anything else like that. But it is just a waste product. Light is a waste product. But what is important in that pathway that I showed you, along this pathway of light production, there are very important antioxidant molecules that are produced. So what is important is the biochemical pathway to produce antioxidants. That's what the fungus is selected for. And giving off light is just a waste product in developing some of these antioxidants. And there's good evidence for that as well. So the take home here is that there's not one reason for why they glow. It depends on the species. It depends on their ecology. But we wanted to test the first one. So what we did was we made some fake mushrooms. These are really cool. So we went to a dentist and got some of the stuff they put in your mouth, and they take the forms of your teeth, right? And we took took these little petri dishes or these little plastic plates. We got the little mushrooms. We made forms of them. Then we took some epoxy, clear epoxy, and we filled the forms. We made these cool little plastic mushrooms. And then we got some little LED lights, and we got the LED lights and arranged them so that they glowed at 520 to 530 nanometers. We took little pieces of wood, drilled holes in them, stuck the lights in the underside of the wood, stuck the mushrooms in the top of the wood, and that's what they look like when you turn on the lights. And then we took these mushrooms and we coated them with a non-odoriferous, a non-smelling, sticky, like flypaper stuff, right? And then we put them out in the woods, ones with LED lights in them and ones that had no LED lights as the control. All right, and we put them in the same woods in the area where we knew these bioluminescent mushrooms grew. And then we came back every day and collected all the insects that landed on both the lit ones and the unlit ones, and then tested whether or not the insects were more attracted to the ones that gave off light than the ones that didn't give off light. And it was really important to do this with non-odoriferous stuff because you have to prove that they're not being attracted to mushrooms because of smell, right? That they're actually seeing it. So we know the insects can see in those wavelengths, and we know that they're coming to them because they come to them and they get stuck to it, and we know what they are. So then we put these things together. I don't want to go too much into it, but basically we tested green light first at two different sites. One site where we had the beautiful little Mycena lucentipes and Lux eterna, and the other site where we have a bigger Omphalodus-like thing, Neonothepanus gardneri, central Brazil and southern Brazil in the Atlantic forest habitat and in a palm forest habitat down here. And then we had collected a bunch of data to show, whether they were attracted to them or not. And then we did some subsequent studies over here, and you can't see it underneath that picture of me inside you there, but basically we tested them in a new moon when it was dark skies, and we tested them in a full moon when there's light skies. We also tested blue light, red light, and our green light. And in all of these tests, the take-home message is that all groups of arthropods are more attracted to luminescent mushrooms than they are to non-luminescent mushrooms. So we know they're attracted to them. So the next part is, do they get spore masses on them? Did they carry those around? Do they actually aid in spore dispersal? We don't have data on that. And so we took it a little bit further, and this will be published soon. Basically, the arthropods during the new moon are 63% higher than the full moon. So more of them could see the light when it's darker out than they can see the light when it's lighter out. So they're attracted to them there. They like it when it's a little bit wetter. More of them show up when it's wetter. They like it when it's a little bit warmer. They like it at a time when there's more insects around than when there's less. So you got to time it to the reproductive cycle of the insects. And they preferred green light to blue light over red light over no light whatsoever. So that was pretty fun. So the future of this was working with the Russians. We'd figured out using Neonothepanus nambi, which is a slightly different species from Neonothepanus gardner from Brazil. Using this species, we took out the enzymes the genes for the enzyme luciferase and the gene for the enzymes for luciferin. And the Russians put them into a petunia. And the petunia now emits light 24 hours a day. And their FDA has approved these for sale in the United States. You can buy them now if you want, as of January of this year. And they're not cheap, but you can buy a petunia now and use it as a nightlight in your bedroom if you want to. So you can grow a house plant that emits light 24 hours a day with fungus genes in them. It's pretty cool, right? All right, so I put this in because I wanted to tell you how important it is to do these sorts of biodiversity studies. So I was really lucky in my career to get the National Science Foundation to fund most of this research, to document the diversity of mushrooms throughout the Hawaiian Islands, throughout Indonesia, throughout Micronesia, throughout Malaysia, throughout Thailand, etc. And we chose areas where there was very little work that had been done. We didn't know much about them at all. And then we went and spent a month or 2 months every day out there collecting. And this process is you go out for 4 hours and collect, and then you bring the stuff back to a makeshift lab, which is a hotel room or an apartment you've rented or something else like that. And you spend the rest of the day until midnight taking photographs, drawing them, writing notes on them, tasting them, taking cultures of them, putting some in little Eppendorf tubes for DNA, et cetera. It takes about a half an hour to take descriptions of a single mushroom. And you do that for a month on end. And if you're lucky, after about a month, you can come back with 100, 125 specimens, something like that. And you do that year after year after year. Well, to give you an example of that, we did some work and only two expeditions in April of 2006 and April of 2008. I went alone in 2006, and Brian came with me in 2008 to the islands of Sao Tome and Principe off of West Africa. Now, this is what we like to call the center of the world, because basically the island is at 0 latitude and 0 longitude. Really pretty cool. So that's the center of the world. Before we did the background research, before we went and did these expeditions, only 4 species of mushrooms were ever reported from the islands. 4 species. Obviously, there's more than that. So we went, and in these 2 trips, we discovered 225 species belonging to 91 different genera of mushrooms. Now, these aren't, I hate polypores. These are no polypores in here. These are all just fleshy mushrooms, just agaricales and coral fungi and puffballs and things like that. So I stayed away from the polypores. And 36 of these we described as new species. And when we worked, and we worked through one group at a time, kind of one lineage at a time. And when we did intensive studies on them, almost a quarter to a third of each of these lineages, what we found in these islands were new to science and endemic to that area. So you can just see just this little bit of work, just a couple of months worth of work here, tells us a heck of a lot about these islands that wasn't known before at all. So these sorts of studies are really, really important. And thankfully, the National Science Foundation has been funding this type of research for at least the last 30 years or so, which is good. And they continue to tell us that they intend to continue to fund it. So as long as we have the general public realizing this is important, that we understand what these organisms are, particularly these fungi, you got to be there at the right time. It's serendipity, as you all know, when you run into a mushroom. So it takes a long time to generate these good data. Okay, now I want to just finish up here, just about finish up here, with kind of how we express fun in fungi. And I want to show you an interesting mushroom here. This came from Sao Tome in West Africa. I discovered this first in 2006, but that was a whole tragic story with that whole expedition that I will tell you. If you want to know, you can ask me later. But we went back in 2008, found it again. Here's what the mushroom looks like, and here's exactly how it grows. Okay, it's a little mushroom here. It's a stinkhorn. It belongs to the genus Phallus. You can probably see why. And it's only 1 inch long. Really, really tiny. It grows on the sides of the logs. It's basically, now it's the second smallest species. There's one even smaller than this called Phallus pygmei. But this is one, it's pretty small, 1 inch long. It's pencil thin, and it curves downward at maturity. Now, when we published it, it made the cover of Mycologia, but the editor said we could not publish it like this. We had to publish it like that because this was too suggestive, right? Well, as it turns out, I decided to name it after a friend. Named it after Bob Drews, and this is because Bob led the expedition to Sao Tome. And he always said, Dennis, you got to name a mushroom after me. Got to name a mushroom after me. So I figured, why not? Let's name the smallest known phallus that curves down at maturity after you. So this is called Phallus drewsii. And it actually, and Bob loves his picture. He says, that's a picture of me holding my phallus. It's right there. This made Wait, Don't Tell. My friend Peter Sagal picked it up, and it made the Bluff the Listener, which they didn't guess, of course. So, and guess what? This made the top 10 award in 2010. This was actually the second mushroom ever chosen to be a top 10. First one was a bolete from New England. But we came in, and so we were able to get the top 10 award 3 years in a row. So I'm actually most proud of that. That's pretty cool. So Brian and I described this one, and yeah, as soon as we published it, it had like 800,000 hits online, most of them porn sites, which of course I didn't visit and get those. And also, there are other ways to do it by besides naming things after your friends. Here's one we discovered down in Mexico, and it reminded us of the spiky punk hairstyle, so we called it Mycena punkissima. This was one that we found in Sao Tome that reminded me of these Kangol flat caps, right? So I named it Crepidotus cangoliformis, formed like a Kangol hat. I sent a copy of the publication off to the makers of the Kangol hats, hoping they would send me a free hat or anything. They didn't even acknowledge getting it. When we published Squarepantsii, Mattel sent me a stuffed toy. They told me they were going to send me some original art because the guy that you probably know, the artist for SpongeBob SquarePants, was a marine biologist who graduated from Humboldt State. And he did that kind of stuff. And he has all the beautiful cells, for the cartoons and stuff. I was expecting an original cell, and I got a stuffed SpongeBob. Anyway, so that was fun naming those. And then, of course, this one in the upper right here is one that I named after my wife, which you have to do, right? And so this beautiful little mushroom here, she discovered in Hawaii on a vacation, because I can't stop looking for mushrooms even on a vacation. She won't let me forget that. But if you look at the etymology of this, which we're required to do, it says, the beauty of this mushroom is surpassed only by that of the discoverer. Now that's the way you get brownie points. It's forever in the scientific literature. It's there. So this is Pseudobiospora leucopatii. And often as well, we try to honor the native folks in the areas where we found their mushrooms. So when we worked in the Hawaiian Islands and described a lot of new species in the Hawaiian Islands, we used a native Hawaiian language. We got together with the Hawaiian elders. They've got a council of Hawaiian elders on the Big Island. And we told them what it was. We showed the pictures of it. We said we would like to have, a Hawaiian name for this mushroom, but we don't want it too alliterative. And if you know Hawaiian, they've only got 9 letters in their language. So you get upa, upa, aka, uka, ua, aoli. That's not going to fly. You have to be able to pronounce it. So we had them, this one on the left, that we named it Lama Lama, which means to glow as if touched by the sun. They're very poetic as well. And the one on the right is called Noa Lokalani. And this means the pink rose in the mist. In Thailand, thanks to my wife, who's Thai, we named a number of species using the Thai language. This is a beautiful ruby red Crinopillus. Tabtim means ruby colored, so it is Crinopillus tabtim. And down in Indonesia, we did a lot of work down in Indonesia. Bahasa Indonesian. Peyong means shaped like an umbrella, and this is Salasobi Peyong. So it looks just like a beautiful little umbrella. So that was fun. And you got to be careful when you're doing this work in the tropics. As you can see, you never know what you're going to run into. Sometimes the locals are very good about telling you what to expect. So here you can see, tiger zone and an elephant zone and things like that in Thailand. Jaguar zone down in South America. But leeches are everywhere. That's a 4-inch scorpion. This is a necrotic spider that bit me in the elbow here and the tissue started to die and fall off at one point. And lots of viperous snakes. And of course, just because there's a coup in Thailand doesn't mean you're going to stop your field work. You just hang out with the soldiers who come around as your guards. We did that. So that's kind of a lot of the mycology. This is the new direction that I've been headed in since I retired. I signed on with a company called Sempera Organics. I'm their chief mycologist, and my role there is to be the scientist to assure that any proclamations that are made are based on real science, not pseudoscience. And if you look around the nutrition industry, the nutraceutical industry, and even a lot of the food industry, a lot of what you hear and a lot about what you read is more pseudoscience than it is real science. But there is a lot of real science out there as well. And now, since there's more funding to do it, there are more human clinical trials that are happening with a lot of different fungi than we ever had before. And that's because human clinical trials are very, very expensive. All right. But now we actually have more resources to help us do human clinical trials. So we can do the in vitro stuff. We can do in vivo with mice and rats. But does that actually extrapolate to the benefits of fungi in humans? You need human clinical trials to do that. So I signed on with them as the chief mycologist. And when the CEO headhunted me and came up and said, our mission is to feed and heal the world with mushrooms, figured I could do that. It just sounds like a good mission. So this is what I've been doing with them for the last 3 years. We got two main things that we're doing. We actually have 3 cores, but I'm only going to tell you about 2. One is nutraceuticals. So these are functional fungi, your typical reishi, lion's mane, cordyceps, all that kind of stuff, right? We're growing those. And I will tell you about one of them. We had our lion's mane, a special lion's mane that we produced with both the fruiting bodies and the mycelium. So not just the fruiting bodies, but both the fruiting bodies and the mycelium. And this is important because most of the lion's mane, most of you might take for cognition. So there's some research out that shows that it can enhance various aspects of cognition. Most of that research is done with cognitively impaired folks, Alzheimer's, Parkinson's, etc. But with as it turns out, there are 2 groups of compounds in them. One are called hericenones that are formed only in the fruiting bodies. The other is called erinacines that are produced only in the mycelium. So this whole argument about fruiting bodies are better than mycelium is really a misguided, disingenuous argument, because there are certain compounds in the mycelium, there are other compounds. Both of them stimulate nerve growth factor, okay? But only the ericinones that are produced in the mycelium cross the blood-brain barrier and get into the brain and can then affect nerve growth, nerve connections in the brain. Right. So what you want, if you're going to take lion's mane, is you want a product that has both fruiting bodies and the mycelium. Okay, so that's what we're doing. We had our product DSX1 and undergo human clinical trials last year out of the UK, and these were done with healthy young adults. The first trial ever done with healthy young adults in humans, as opposed to cognitively impaired. And the results of the trial were very positive. It showed that reduced stress in healthy young adults. Who doesn't want that? And it increased the speed at which you do cognitive tasks. So you can think a little faster, you can remember a little bit faster. That's pretty cool. We now have a second trial. It is undergoing right now. It's in the progress right now. Hopefully we'll have results by the end of December, also in the UK, but this is done with healthy young women. 18 to 25. I'm told that they have a lot of stress. So that's a joke. So this trial is really going to be important. So if we can show that it actually reduces stress as well, this is going to be very significant. So we're hoping for that. So that's one aspect. The other aspect is we have a new food that's out there. It's called Mamu. You can get this by going to about 15 different restaurants in the Bay Area, or it's throughout hospitality in Las Vegas. We're trying to take it nationwide, but that's going to take a while. This food, Mamu, is made from 3 species of mushrooms and mycelium. So the mycelium of king oyster mushroom and lacto-fermented shiitake mushroom and white button mushrooms, and that's about it. Very clean label, very pure, processed in a various low processed way. And then it looks like we add beet powder to it, so it's red. It looks like hamburger. It's a ground product, and you can make anything you want out of it. It's absolutely fantastic. So people are excited. We're actually showing it to McDonald's in 2 weeks. They're excited about it. They don't like Impossible, which is soy protein based. This is all mushroom based. So basically, we're trying to promote this as a new pillar of food. You got your plant pillar, you got your animal pillar, you got your fungus pillar. So people need to eat more fungi. It's a complete protein. There's no gluten, low or no fats, on and on and on. Just very, very helpful product. So that's kind of what I've been doing, and that'll take me up until I croak. And thank you. Yeah, there. I had to put that out because this was 2007. I'm going up to Northern California next week. I'm with Ann and we're hoping to find that. I know they're coming out. So if you have any questions, I'll address them, and if not, thank you.
I had a question. Those cells that come off of the mycena, are they the asexual propagules? Are they dikaryotic?
They are.
So is
That grows directly into a mycelium that can produce mushrooms.
So
They're total clonal dikaryotes. Yep. Yeah.
You said with the Ricci and the mycelium is able to the chemicals in the mycelium, the compounds can cross the blood-brain barrier.
That's with lion's mane.
Yeah, the Ricci, right?
Oh, I thought, sorry, I heard
Yeah.
Rishi, sorry, yeah.
Yeah, my bad. Maybe I did say Ricci, I meant Ricci. In the fruiting bodies, is that able to affect peripheral nerve growth?
Yes. So that's it. So if you, my problem with a lot of the products that are out, if they're selling lion's mane, they say it's all mushrooms, don't buy any of the stuff with mycelium in it. It's all mushroom because it's really good for cognition. It's not really that great. There's no data that shows it's great for cognition. It's great for stimulating nerve growth factor, and we have nerves throughout our whole system. So for endurance, stamina, things like that, and movement, muscle, stuff like that, it'll definitely help that throughout the rest of your body. But it's not necessarily great for cognition. But the arenacenes in the mycelium, a lot of good clinical data showing that.
Has anyone done any work with like stroke patients or anybody who's like had peripheral nerve issue from whatever?
Not that I know of yet. We've got a guy, we have another trial in a different UK at Cardiff Metropolitan University, who studies concussions and sports injuries, things like that. And he's studying it right now. We just got preliminary data on that, and he's really excited about the data from that. So I want you to stand. There you. Yeah. So yeah, that's another good area to look into as well. Yeah, Ken.
Of all the mushrooms, all the luminous mushrooms that you investigated, question is, which is the brightest and the easiest to cultivate? Like if you wanted to actually grow your own light bulbs or whatever, figure that out.
Yeah.
For the future, what's the best potential to work with? Maybe top.
Okay. All of those that we have tried, and we've cultured a lot of them now, the mycelium grows like crazy and it glows like crazy. So you can fill petri dishes with mycelium. If you want to get them to fruit and actually have the mushrooms, that's a whole nother question. So to answer the fruiting question, for mycelium,
I'm not so interested in the fruiting if it's going to glow better in the mycelium. I just, who's the brightest mycelium or mushroom or whatever?
the brightest that I've seen is Neonothopanus gardneri. Both the fruiting bodies and the mycelium. It's really pretty bright. I mean, you can put the mushrooms down in a totally dark room on a newspaper and read the newspaper. Okay, but they're also, it's not an, Omphaliotis olearius, the jack-o'-lantern mushrooms, they don't really glow that bright. They're pretty dim, but they're big. Well, this one is in that lineage. It's a sister genus to Omphaliotis. They only grow to, maybe about this big, which is pretty good size. And they look just like an Omphaliotis. They look like a chanterelle, really. The current gills, chunky. And that thing glows pretty darn bright. And we've actually fruited it once by it grows on palm debris. They're taking palm debris, autoclaving the palm debris, making good substrate out of it, then inoculating that. The mycelium grows all through. You take that bag of palm debris. We did it like in a big garbage bag, a translucent one, took it in a dark room, and the whole thing's just giving off light. It was pretty cool, but it actually formed some fruiting bodies on it as well, which is pretty neat. But easier to fruit, if you want to fruit the mushrooms, is Mycena chlorophyos. You can get the cultures of that. It'll fruit right on a petri plate, but you can put it on all kinds of other media, and some of those mycenas will do that for you.
Follow up to that, a few years ago, you had come around to a society meeting, well, I remember you asking about if we had ever found any of the local armillaria species to actually be as glowy as the one back east.
Right.
Have you ever found the foxfire
Yes.
armillaria?
Yeah.
And is it really, I always hear these legends of fiery waterfalls when you dump a log over a cliff or something, it explodes on the way down.
The mycelium of our local armillarias does emit light, because I've taken it and done it on petri dishes in the lab and shown that. If you sit in the closet by yourself in the dark for 10 minutes, your eyes will adjust enough to where you can see it. Right? If you take a digital camera, you get a good photograph of it in no time, right? But it does glow. The fruiting bodies don't. But now we've discovered 2 species of armillaria that we haven't, Brian and I have been trying to put names on them for a long time now, still in the works. 2 species of armillaria from Brazil, where the fruiting bodies glow as well as the mycelium. And that's really some of the first armillarias. Taylor Lockwood has photos of one armillaria from somewhere where the fruiting bodies are glowing. New Zealand, excuse me, that's where it's from. New Zealand, yeah.
Have you ever figured out which one back east it is that has that legend?
Oh,
Waterfalls from the log.
I don't know where that legend, I don't know where that legend comes from, but the mycelium of all of those armillarias back east emits light. The rhizomorphs don't, but the fan mycelium does. And it grows between the bark and decorticated wood. You pull the bark off and you'll see it, definitely. Yeah.
What led to the discovery of the first aquatic mushroom? Why were you looking?
Yeah, I wasn't looking. I have a colleague down in Argentina, Mario Reichenberg, and he had a graduate student who was studying sedges. So he's a mycologist, but she was studying pathogenic fungi on sedges. And so she was doing her work up here at that lake. And why she was collecting and cracking the ice open and pulling out sedges in the winter, maybe she was looking for tolerance of her pathogens against it or whatever else. She pulled out the sedges, saw these little tiny mushrooms on them. Mario calls me up because he doesn't study. He studies polypores. I don't. And called me up and said, "Hey, we got this thing." And so we worked on it together and published it. Yeah. A lot of this is serendipity. I mean, that's the way you find a lot of these things. That first bioluminescent fungus was a chemist out there finds it, and it ends up in and now we know twice as many. Yeah. All right, thank you. It's been a lot of fun.
Question.